turn it over to Adam, and so he can share his screen with you and get started. Thank you, Adam.
Thanks so much, Jay, and it's a great privilege to be here. You guys can see my slide deck, hopefully. Let me know if not. And as Jay mentioned, we have been partnering closely with Casey and his team at the Data Lab, and today I wanted to go through really the odyssey of our efforts, not just right now in the context of the Data Lab, but how we got here, where we're trying to head, and how we're trying to really iterate on new models of discovery and accelerated research, particularly in the context of pediatric brain tumors. So pediatric brain tumors really present a tremendous challenge on a global scale. The landscape within the United States really is such that, and most people don't recognize this, but pediatric brain tumors are the most common cause of cancer-related death in children. But this is true in a variety of different contexts that span into the young adult age. There are more than 4,000 children and young adults are diagnosed with brain tumors every single year. The median age for across these patients is only 9 years old for those who succumb to their disease. And in totality, on a yearly basis, there are more than 14,000 children that are currently living with brain tumors. But this is not just a US problem, and it turns out that this primacy of leading disease-related death in children is shared amongst the entire world's context, and every country you see here and many more are facing the same challenge. And as such, this really creates a global challenge, and as challenging as it might be to within the United States, it's even more challenging in many other places to really drive change and impact on behalf of children who are suffering from pediatric brain tumors. And so for us, Open PBTA is really an experiment. And I say that in ways that perhaps is a disclaimer, in that it's still an ongoing experiment, an ongoing initiative in which we're continuing to iterate on how it is that we can change that landscape, both here in the United States and beyond our borders. And the challenge really centers on this concept of an experiment. I was trained as a traditional scientist, really as a neuroscientist. I did my graduate training in very traditional fashion at Johns Hopkins, and really began as a PhD, searching to drive change in the context of pediatric brain tumors. And this is the model of research that I was really trained in, the scientific method of creating observations, testing hypotheses, performing experiments, and iterating on the conclusion that those experiments drive. Here you see Galileo's Leaning Tower of Pisa, where essentially he's testing his hypothesis on the effects of gravity in ways that are invariant of shape. And so this is really not unique to those of us in graduate school, but whether you're in first, second, sixth, 12th grade, this concept of how to do science, as driven by the scientific method, is drilled into you from the very beginning. And for those of you who don't recall, I'm sure pictures like this might provide some memory. But it's actually very challenging to simply take these sets of boxes and implement them on behalf of pediatric cancer and pediatric brain tumors. And so this is me and the ID badge that actually I still carry from the Children's Hospital of Philadelphia and my arrival here in 2007. And I was a very naive scientist at that time, coming to CHOP, really wanting to do research in this fashion. But at the time, I rapidly faced a challenge, which was that at the time there were no large-scale NIH initiatives focused on brain tumors, and it was very challenging to even begin to ask the right questions or form the hypothesis or do the experiments. And part of this challenge is that although pediatric brain tumors are the leading cause of disease-related death in children, thankfully most children are healthy. And so pediatric brain tumors and most cancers in children are still classified as rare. Now, rare is an arbitrary term, but in this context, what it really means is that even at a place like CHOP or any other hospital system, it is very challenging to get access to sufficient number of specimens, sufficient number of data sets or tissues to perform these initial steps of the scientific method. And so I actually also ended up being an English and literature major in college. And myself and working with several partners at CHOP and then other institutions began to really think about this scientific method challenge that we faced. Very much aligned to T.S. Eliot's sort of prescient characterization of something that's very close to the scientific method, but recognizes the difference between knowledge, information, data, or here in this context, wisdom. Specifically, this actually ended up being, T.S. Eliot actually wrote this in 1934 before we even had ways of measuring information. The concept of the bit essentially didn't come along until later, mid-century. But this concept of data to information to knowledge to understanding actually became fairly well established, especially in the 1980s, as a methodology for thinking about the flow and the handoffs that are required between these concepts of data, information, and knowledge, and what it really means to transform data into information and information into knowledge and its use. And as we began to iterate on this concept, we really began aligning that methodology to the scientific method. And it was clear that in order to create observation, hypothesis, and experiments, we had to solve this data or information or knowledge gap if we actually wanted to get to impact, if we wanted to actually translate efforts into that context. And so to provide some context for the definitional space, data is really raw data. So you can imagine here in this context, sequencing a tumor and generating the first genomic sequence from that tumor. And that genomic sequence, in order to actually be interpretable or queryable, you have to harmonize that genomic sequence to other sequences. You have to apply some standards to it that initially allow you to penetrate really what is in that sequence. That on its own does not provide you knowledge. Knowledge is really taking that information and going through the process of experimentation or analysis such that you can interpret what it means to have that sequence. And then obviously the impact is the space where you then can act upon that meaning on behalf of altering the course of the biological context. But above this pathway, you can see here something that I alluded to earlier, which is any one scientist at any one institution that's bounded by their laboratory or their institutions has a tremendous challenge generating enough data to actually transform it into meaningful knowledge, in part because we have to think of this as a global challenge. And so if you actually want to even perform experiments, and then secondarily, if you layer on that the mandate to actually accelerate the impact that you're trying to get to, you have to really reconceptualize how to do this process in ways that I think, ALSF and Nate Foundation, the Data Lab, and many others are really driving ultimate change. And here I just have, the equation for acceleration. I always remind myself that it's really, if you want to accelerate, by definition, here, Newtonian, you actually have to drive change or change the velocity across the time. And critically, time is the variable that in the translational context, if we want to have meaningful impact on patients, we have to optimize for. So the question ends up being, how do we drive this impact in the shortest amount of time with limited local resources? And so this is where the birth of the Pediatric Brain Tumor Resource and Atlas Initiative really began to form, with the idea that we need to think of patients beyond our own hospital system, to think about a network of hospitals that have to be connected in near real time to platforms that support that data to information process, and that this has to then be further underpinned by human partnerships that harness knowledge and expertise across very broad spaces of scientific knowledge. Specifically, throughout the entire process, and every patient family knows this, when you are dealing with, let's say, pediatric brain tumors, even as a family, you end up spanning a whole set of expertise with highly specialized people who have spent their entire lives learning and improving the application of radiology or diagnostics or surgery or chemotherapy. But it's really putting those systems together that drives the ultimate care of the patient. And likewise, on the scientific side, it's really bringing together very diverse expertise, from the molecular biologist to the genomicist to the proteomicist to the data scientist to the clinicians, all together around a patient's data-driven care that requires us to coordinate these activities if we are to actually drive that accelerated impact. And so the Children's Brain Tumor Tissue Consortium, as it was first known, more recently it was transformed to the Children's Brain Tumor Network. So you'll see both actually highlighted, both names utilized throughout this presentation as we're rolling out the change in name. And it'll make sense as we move forward. Really began as initially 4 institutions that really wanted to drive that biospecimen to data generation context. And we began the consortium in 2013, initially with 4 institutions, and today we span almost more than 30 institutions. And what I'll walk you through is really this timeline of efforts, and then point you to some of the resources and products of these efforts in ways that we hope to engage you and the rest of the community in partnership, even beyond this initial consortia-based efforts. And so we launched a consortium in 2013 with the idea that if we could actually bring together as a centralized resource source the biospecimen, associated clinical data, the longitudinal clinical data, the MRIs, the pathology slides, we could create that data resource such that any scientist could come to work on pediatric brain tumors. And that was the goal. In fact, that's a major challenge still today, that there aren't enough pediatric brain tumor scientists in general to solve the myriad of challenges and across the myriad of histologies that exist within pediatric brain tumors. But 2013 was a start. And today there are more than 21 institutions who are part of the Children's Brain Tumor Network that span essentially the United States across both coasts, as well as in Europe, Asia, and Australia, all committing to the development of this resource as a global resource. So, and what I mean by that is that the specimens and data sets are not only available to the institutions who are contributing those, but to anybody who actually wants to use these specimens. And over the last several years, this specimen and biorepository and data resource has further expanded its relationships with clinical trial networks, especially with the Pacific Pediatric Neuro-Oncology Consortium, where large-scale data sets and biospecimens can drive the preclinical work and clinical trial development area such that they feed into new clinical trials and vice versa. Those clinical trials generate data sets and biospecimens that feed back into the consortium context, creating what we really think is the world's largest pediatric brain tumor resource supporting research. So today we have more than 3,000 patients enrolled with longitudinal clinical data in the Children's Brain Tumor Tissue Consortium or Children's Brain Tumor Tissue Network. This spans more than 30,000 biospecimens that essentially span fresh frozen tissue, DMSO frozen tissue, so you can generate cell lines or preclinical models, as well as derivatives of the tissue like RNA and DNA and protein for various assays. We've also been curating and developing preclinical models with more than 70 of these. These end up being the environment or the resource that allows for drug testing or CRISPR knockouts or any of the other types of efforts that really drive accelerated clinical translation. And the Brain Tumor Tissue Atlas initiative really was spawned by the sequencing of 1,000 of these more than 3,000 patients. So it's only a little bit less than a third of the patients that are currently enrolled that are characterized, but 1,000 whole genomes and RNA-seq sequences represents, to our knowledge, one of the largest molecular data sets ever created on behalf of pediatric brain tumors. To date, I think there's less than a couple hundred patient-specific data within NIH resources. And so this really represents a tremendous community-led effort to drive the generation of large-scale data. These have then intersected with NCI and NIH-based efforts to layer on top of the whole genome and RNA-seq context proteomic data, as well as very soon single-cell or single-nuke RNA-seq data sets, creating really a very rich multi-layered data set for these patients. And for us, this began addressing that first challenge of data. How can we move from biospecimens to data as a starting point in that value chain on behalf of creating information? And again, at each step, I have to be transparent, although as you prepare a presentation, you put it together on a slide deck that makes it seem like you preconceived and designed this rationally. There's always an iterative process in optimizing how we go from collection to annotation to curation to QC to generating these data sets and then ultimately driving the informational context. It became clear that this more than 3 petabytes of data that was generated was an incredibly rich resource, one that exceeded the capacity of CHOP or any one institution just to analyze. And so very early in, we began looking at how do we create cloud-based infrastructure that can support the access and use of these data sets. And I'll just briefly highlight a couple of these to show you where things stand and how anyone can access these data and connect that to the concept I'm trying to elucidate here and the still remaining challenges and opportunities I hope the audience will further engage with us. So Cabatica is a cloud-based workspace environment. So each time you generate a whole genome or any sequence, in order to convert it from data to information, you have to process that. And this requires significant compute and analysis and workflows. And if you actually even want to recreate or reproduce these initial processes, you have to implement those same workflows in a routine way. The Gabriella Miller Kids First Data Resource Portal is actually an NIH-funded environment that not only hosts the Pediatric Brain Tumor Atlas datasets, but importantly, many other datasets. And that's a critical factor that I'll get to later. And that is that in any discovery effort, what we have seen is that in order to maximize the interpretation of that information that you're trying to generate, you need to contextualize it in as broad a biological context as possible. And then we partnered with Dana-Farber, Princess Margaret, and MSK to further iterate on a very often used environment called CBioPortal, which really is an environment that takes the output of activities in Cabatica, the processing of that data, and provides a way to query that initial process data. And I'll show you that briefly. So let me switch screens just so I can highlight some of these platforms, largely to provide you a context for how we're trying to solve that data to information phase. So the Kids First portal is an environment that any user can actually log into, and this is myself. And when you first log in, you'll come to your own environment, and you can explore any data set that's in the Kids First portal. But as I mentioned, we were able to leverage the building and construction of the Kids First data resource and really partner with the NIH to iterate on how to optimize building a portal environment to allow users to initially identify data sets around pediatric brain tumors. And so you can see the Pediatric Brain Tumor Atlas is available here. And this is that first 1,000 or so subjects that I described coming out of the CBTN. And if I apply that filter, you can immediately see that there are 917 participants with a whole variety of different phenotypes, different data modalities, both RNA-seq and whole genome data. And you can see some of the other metrics like demographics, the different histologies, different ages, and so on. The portal is meant to support users who want access to the source data. So what this allows users to do is to create essentially your bag of data that you might want to use and analyze. So users could also, for example, start a new query and say, I want to not only analyze brain tumors, but I'd like to also analyze neuroblastoma. And then I would like to go to these more than 20,000 files that are available across neuroblastoma and brain tumors, and then further iterate on the types of files and source data that I would like to analyze. So we've had people who essentially analyze the raw genomic data sets, people who are interested in gaining access to processed data that are available, or other file formats that include, for example, the slide images from pathology or the PDF reports coming from the hospital system, so the pathology report or operative report for various analyses. This environment with your bag of data is a place where many users begin, but it's especially geared to one type of user, and that is the bioinformatic analyst, which, to be honest, is not me. Remember, I was trained in a neurosciences, so someone like myself would be very challenged to start here and go all the way to the information and knowledge space. And that's the first important lesson in this context, is that it really requires combinations of teams. Yes, I, as a scientist, can create the bag of data, but typically I would have to partner with other users and or experts to process that data. However, I could take that data here and create an environment where I could analyze that data. Oops, I think I clicked on the wrong ones. Sorry. Where I could actually analyze such data in an environment like Cavatica. So here, I'll just go to the Cavatica environment here. Now, once you click on the user data that you're interested in working with and analyze the data in Cavatica, you're brought to an environment where you can actually have projects that within those projects, you can actually invite your favorite bioinformaticians to come and work with you on that data set. And so I, as a biologist, can say, hey, I found this amazing data set, but I'd like to work with my favorite bioinformatician and invite them to come into this workspace. And furthermore, this is also the place where we began creating public projects, which I'll come back to at the very end, but wanted to highlight now that the open access data sets are also available here. And I'll come back and reference them later. Someone like myself, as I mentioned, would begin as a scientist or a biologist to query data sets after they've been processed in that initial space in an environment like cBioPortal. Here, you can apply your domain expertise, look for your favorite gene. So this is, again, the Pediatric Brain Tumor Atlas initiative, where all the process data, this initial provisional process data, is available. And I can look to identify all the patients that might have a P53 mutation, and then look to see what other mutations they might have or other features of that cohort in a meaningful way. Let me come back to the slide deck. So in many ways, and again, from a naive perspective, our consortia-based efforts and initiatives really thought that this is what it would take to drive change, that delta, that we began generating large sets of data, created environments where users could access the source data, could process that data, created initial environments where people could peruse or ask questions from a gene-centric view or patient-centric view. But the reality was that it did drive that process in a significant way, but to an extent that we want to drive further. And I'll give you an example here. So for us, it was phenomenal that we've had, over the course of less than a year, more than 100 investigators essentially access the raw genomic data sets. More than thousands of investigators began using the process data. And more than 60 investigators actually applied and received samples from the biorepository. And again, these numbers for us as a community are phenomenal because, there are less than 100 probably pediatric brain tumor investigators or PIs in the United States who are focused solely on pediatric brain tumors. So having these types of numbers is really phenomenal. But when you start thinking about the number of different histologies that you're trying to address, and you can see this here, you start recognizing that if you start splitting these investigators across the different histologies, it's still a small number of investigators. And our goal should be not just to serve those who know they want to study pediatric brain tumors, but to expand this community to hopefully some of you who are on the call and beyond that context. And the way to do that is to drive knowledge creation. One way that I like to characterize it in a sort of an analogy is that when any one of us is creating a meal, if we were forced to go and fetch the eggs from a chicken to cook our eggs, very few of us would be eating eggs. And so, for us to actually cook, most of us, I suspect, to cook a meal, right, we rely on a whole value chain that gets us the eggs in a simple way, that then we can cook the meal. And we can iterate on the meals that we cook. You don't have to be an egg aficionado, and there are people who are like that. There are chefs who are experts, but even non-experts can cook eggs if you make the eggs available to them in an easy and approachable way. And this is what I'm trying to hint at, that even as you lower the bar for access to raw genomic data, you're still required to work across a very diverse landscape of investigators to continue pushing that data to information and ultimately to knowledge. And this is really where we found keen partnership and opportunity with the Data Lab, as Jay mentioned. Because it turns out that going from data to information to knowledge to impact is not as simple as T.S. Eliot thought, and it's not his fault. And that's because each one of these spaces, these lines that separate data to information, information to knowledge, and knowledge to impact, one, they're not unidirectional, so they're iterative. But two, almost always there's a change in domain expertise and the kind of people and their specific knowledge, even independent of a specific disease, that's required. And so it's really a digital and non-local community that's connected through platforms and performing the actual knowledge creation together that will optimally drive change in the time variable of interest. And this is supported by platforms that integrate across these efforts, but also a community-based approach to knowledge creation. And so OpenPBTA is an experiment, but really it's a new way for rapid knowledge creation to take that information context and drive iterative knowledge that can be a starting point for expanding the network of those who actually intersect with that knowledge. So you don't have to be a brain tumor scientist or a pediatric brain tumor scientist to intersect with that knowledge, because knowledge spills over across different domains and disease contexts. And so this is where the Open PBTA initiative and our partnership with Casey Green and the Data Lab really had its genesis. And in part, it's because of the Data Lab's sort of visionary approach to thinking about knowledge creation. Specifically, and this may have been introduced in some previous lectures, but specifically what they have done is brought to the knowledge creation context the open software context, leveraging really GitHub. And for those of you who are not GitHub aficionados, it's actually a fairly intuitive environment. And to be honest, most basic scientists or biologists don't intersect with GitHub because it's a software development arena. But a truly intuitive way that does that iterative cycles I mentioned before, that iterative cycle between information and knowledge in a way that's very thoughtful. And so here you can see this schema where a project, in this context, the Open PBTA, is created, and where various users are intersecting to create knowledge through specific analyses. And those analyses and their outputs are iteratively integrated through a context of what's called a pull request, and then emerging into essentially a master branch, as it were, of the analysis. And I'll go through that in just a second. But this complicated graph, all it's really doing is saying, I'm going to harness the expertise of different users who are interested in performing different analyses to drive new knowledge creation, but do it in a coordinated way, in a transparent and reproducible way that can then intersect into a combined knowledge space. And to my knowledge, this has not been done before in this way when it comes to pediatric cancers, and likely very infrequently in the context of almost any other scientific endeavor. Again, historically, we work within our institutional boundaries, and even if we're willing to share data, that sharing is a transactional activity as opposed to a collaborative activity. So, for example, and I'll show you in a second, when you go to the GitHub Open PBTA environment, you'll see transparently all the proposed analysis, and anybody can propose an analysis, anybody can actually perform an analysis, and then those analyses can be integrated into this living context of shared understanding. And I'll switch screen again just to show you that view. So we can just go to the Open PBTA analysis under the github.com Alex Lemonade Open PBTA analysis, and you'll see right away a list of activities, and right below you'll see more concrete description of the effort, really highlighting our efforts to fully convert these 1,000 or so subjects data sets into meaningful results. And importantly, you can see that what as a community, and it's not just us, are proposing to do. So anybody can actually propose, and I'll pause briefly to thank all of those who may be listening who have proposed analyses on these data sets. And you, at the same time, can actually also look at these analyses and specifically their results. So for example, one analysis here is the immune deconvolution. I can see that Jacqueline actually submitted this issue here from the Data Lab. And the very important part of this process, as supported by the Data Lab, is that the process of doing the analysis in order to integrate into the workflow has to be reproducible. So each layer of code and analysis that extracts meaning from a particular data set. So for example, here, the immune context of an RNA-seq. Not only could Jacqueline run this workflow, but anybody can actually take the exact same workflow and come up with the exact same answer, and more importantly, with the exact same plots. So for example, here, I'll just show you briefly, and you can iterate on this, but you'll see just a brief preview. Here is this initial output and clustering of the immune context based on RNA-seq for the PBTA. This process of creating analyses and figures and knowledge in ways that are fully transparent pre-publication as a community is a remarkably, at least for someone like myself, innovative way to drive that accelerated impact. And I'll show you in what ways I think this is meaningful in just a second. So as I mentioned, anybody can actually go to the OpenPBTA analysis GitHub repo and then analyze it. So I know I went through a variety of different portals and environments, and to try and tie this together, let me just briefly encapsulate these together. So we knew that users would want access to data. You know, we've managed to create an open science repository of biospecimens, managed to at least sequence and characterize to some extent, you know, about a third of these specimens. But we knew that we had to provide a way for people to find and create their bags of data that they might want to utilize. And this is especially true for data scientists and bioinformaticians, as informed by domain expertise working with biologists or clinicians. That bag of data can then be analyzed and processed in ways that convert it from really data to information. That first alignment or even variant call space can be done across domains like Cavatica and then even queried in the context of PHC Bioportal. But if you ask me, how does PHC Bioportal help the next patient who comes through CHOP's doors? That's the space where I would feel uncomfortable, because I wouldn't be able to say to you, how can I position the next patient's whole genome or RNA-seq data in the context of all of the OpenPBTA data set? And that's the important transition that happens once you start analyzing the data. That's where knowledge comes in, because then I could take the immune profile of any one patient that comes through CHOP's doors and position it in context against all of the knowledge created by workflows in OpenPBTA. And I'll show you what that looks like here. So I'm going to switch screen one more time. Oh. Here. Oh, sorry. There we go. So here's actually an environment where we've taken all of the OpenPBTA immune profiles, and they are arrayed here as box plots. So each one of these box and dots represent all the patients in the OpenPBTA. And this red dot is a specific patient, a patient actually in a clinical trial. And so you can take that knowledge space, right, and begin to understand that this patient, compared to all other patients, has different immune cell context. You can see here, right, the adipocytes or the HSC cell content, or even the melanocytes, any of these cell types, as predicted by an immune deconvolution workflow, can be essentially interpreted in context. And once you do that, then a clinician can begin understanding and interpret that specific patient data set. And for me, this is really a fulfillment as a scientist, and I think it's true for every one of us in the pediatric cancer landscape, of a vision of taking a data-driven approach that begins as a partnership with patients who enroll in a biorepository initiative and ends with patients who are counting on us to provide them new avenues for the interpretation of their specific tumor. So, and it's a phenomenal story, and again, I'm highlighting that the Open PBTA analysis is still ongoing, and there are still broken pieces, things that need to be fixed, issues that need to be resolved, branches that need to be merged. But I wanted to highlight this vignette of the opportunities that come to fruition when you engage a community-based approach in more real time, disconnected from the traditional practices of individual investigators or scientists working as independent agents within a particular institution. And the part that I hinted toward throughout the presentation, which is that we need more of those independent investigators overall, not only to work together, but just in general, the total number of investigators itself to work in pediatric cancer also gets repositioned as part of the Open PBTA initiative. I think that for me, this was a thrill only a few weeks old, where essentially, in a meeting that was ostensibly about something else actually, with one of our collaborators at Mount Sinai, Avi Mayan, he actually introduced us to Nicole Mosayev, and she is actually a second-year undergraduate student who was spending time in Avi's lab just for the summer. And like most bioinformatic environments, and Avi's is a bioinformatic laboratory, they often focus, and you can see it here, on the TCGA. This is the large-scale adult cancer data sets that has really transformed the way science is done and where we are trying to model many of our efforts. But Nicole, the minute she found out that OpenPBTA had processed data and matrices, took the work that she began working on in the context of the TCGA. And again, I'm a trained neuroscientist, so I couldn't necessarily deconvolute for you the entire here Jupyter Notebook code. But she began actually taking this Open PBTA context of the data, and immediately, right, by just taking the files that are available as part of the Open PBTA. So this PBTA stranded.csv is a file that's available on the GitHub repo. And she could take that file and begin to actually, and this file as well, right? She could just pull those files, right, using her notebook code, and begin to iterate on the analysis from her own perspective, even as an undergraduate, in ways that was quite meaningful. In fact, very rapidly, and I'm gonna scroll down briefly, she could begin to cluster and explore the data. So here, she created a way to actually, and again, these are all field and analyses that were done under OpenPBTA, but she can actually begin to cluster and look at different tumor types as analyzed and defined by her own methods to further iterate on the knowledge of any one specific patient, but in the context of very many. And this is someone who has never touched or been informed about pediatric brain tumors, but because we brought the egg, right, to her refrigerator, essentially, she's able to cook her omelet and drive further knowledge and interpretation in ways that we hope then feeds right back into the data-driven process of discovery for patients. So again, this is still very much an ongoing experiment, and we hope that those of you who are on the call will join, inform, make us better at doing this, because ultimately it's on behalf of patients. And, we hope that those of you who are on the call will join, inform, make us better at doing this, because ultimately it's on behalf of patients. And, we hope that those of you who are on the call will join, inform, make us better at doing this, because ultimately it's on behalf of patients. And, we hope that those of you who are on the call will join, inform, make us better at doing this, because ultimately it's on behalf of patients. And, as we in our community, and this is true for brain tumors and beyond, we won't stop until every child is cured. And this, as far as I can tell, and I think many of you likely agree, depends on a data-driven approach that is accelerated. I'd especially like to thank Casey and his team in the data lab, especially Casey and Jacqueline and the rest. They have been transformational, not only for our team here at CHOP, but for many others, in pushing and iterating on how to drive knowledge creation through an open science approach and their efforts. And we wouldn't be anywhere near this space if it wasn't for their partnership in this context. And obviously, you know, I'm just a neuroscientist, and all of this wouldn't be possible if it wasn't for the amazing team within the Center for Data-Driven Discovery and Biomedicine and our consortia partners who have selflessly contributed all the specimens in partnerships with patients and foundations who have sponsored most of the data generation I talked about today that is available for use right now. So thank you so much. And again, hopefully this can act as an engagement opportunity, and please follow up with any questions or issues with our team. We hold frequent open office hours around these platforms and can always facilitate access to data or support new projects associated with specimens within the biorepository of the CBTN. Thanks so much.
Thank you so much, Adam. I appreciate the talk, and I appreciate you sharing on how people can get involved with the work that you're doing. We're going to go and see what questions we have now, if you have a few minutes to answer questions, okay?
Of course.
Okay. The first question we have comes in from Hendrik Lim. I know I've seen Hendrik on these talks before, and his question is, what is the rationale you take for doing WGS and RNA-seq data from the same subjects at the same time?
Yeah. So this is not just my rationale. You know, early on, we actually essentially polled our community for what would be the most salient data modality to generate that would drive initial interpretation. And because so little data had been generated or has been generated in pediatric brain tumors, in order to get a comprehensive view of the molecular landscape, we began with whole genome sequencing and RNA-seq, and that has ended up serving us incredibly well. Not only does it allow us to look at the potential mutational context of the tumor in coding and non-coding regions, but also integrate that with the so-called germline context. And then layered on top of this initial genomic static view is the expression or RNA-seq data for each one of those tumors that then allows us to interpret, for example, the immune context. It's also served as a layer upon which other data modalities that are less well harmonized across different groups, but which add additional meaning can be added. So for example, once you have the whole genome and expression data, you can layer on proteomic data or single cell analysis data. And that process has really proved useful in ways that we can then integrate with the molecular characterization workflows that are currently emerging in the clinical context. So the example I gave of a patient-specific analysis is based on the fact that in that clinical trial, there's whole genome data and RNA-seq that the clinicians are trying to interpret. And so it's really trying to link that discovery canvas of the open PBTA to what we know are the modalities that clinicians are using at the front lines when they meet patients.
Thank you. And Hendrik says, thank you for the answer in the talk. The next question comes in from Lindsay, and the question is, how often is new data added to the portal?
Yeah, we actually try to add data in as near real time as possible. So just to give you as an example, that patient that I highlighted from a PNOC clinical trial, that's an ongoing clinical trial that's taking place right now. That data is already integrated into the open PBTA. And there are various approaches to doing this, and there are both benefits and challenges in doing that. Our goal is to provide access to provisional data as rapidly as possible and be transparent about the fact that these are the data sets as they are coming in. These are the data sets as we are processing them. And we want you as a community to work with us in that context. That's a very different model than most NIH studies, which essentially have data release cycle. So essentially, you have a cohort of subjects that is curated, analyzed, processed, and then on a yearly or multi-year context, there's a data release of highly curated data. We chose as a community to work through a transparent process where, yes, there will be a curated, validated, processed data set, but everything along the way is also available. And so we really want to engage that early process of discovery, curation, interpretation as rapidly as possible, because those are actually challenging things. So our goal is to deposit and make data available as rapidly as possible. And as you saw, each time a group requests specimens for use, it's incumbent as part of the process that they sign on to, that when they generate that data, it comes back into the repository. So what you'll see is fairly frequent updates and integration of data, clinical data, and biospecimen-derived analyses into the workflow.
Great. I mean, it seems like in today's environment, everybody wants instant gratification. So you're trying to address that, right?
In part, but in part, we also need people's support, right? These are large data sets, and it turns out that the best way to identify issues with data or interpret them and curate them is to actually use them. And so that's why sort of the Open PBTA has really served us amazingly well. And even the Childhood Cancer Data Lab has been amazing at this. As we iterate through the data sets, we can improve them, curate them, re-annotate them, and work through both a data curation, analysis, and QC process simultaneously, all with the goal of the accelerated discovery process.
Okay, so the next question comes from Amber. And Amber says, we're in the early phase of building a pediatric thyroid consortium. And it'd be great to ask some specific questions related to early building and strategic planning. Is there someone that we could email to set up a meeting?
Yeah, so please email me directly. And this is a very important question. What we found is that there's such value in engaging domain experts with a specific focus like thyroid cancer, or you can even imagine a specific brain tumor like medulloblastoma or DIPG. You need to have a node for that community to aggregate on and commit to and guide in a disease-specific way. But what we found doesn't work so much is for each one of those efforts to try to recreate the entirety of the infrastructure from foundation to the highest brick. It's such an expensive and challenging effort to do in a disease-specific way. And so our hope is that any community, thyroid or otherwise, could leverage the platforms and resources we've created. And so we've tried to make many of these very modular. And at the same time, it also allows not only for groups to develop a disease-focused effort, but secondarily to interoperate across diseases. And there have been too many lessons that we've learned where simply because data sets were not interoperable or interconnected across variegated domains, as an example, diffuse intrinsic pontine glioma and fibrodysplasia ossificans progressiva. These are two diseases, FOP and DIPG, that have ostensibly nothing to do with each other. One is a rare bone disease and the other one is a horrific brain tumor. But 1/5 of patients actually share the exact same mutation between those two. Normally, these communities don't interact with each other. The clinicians don't work with each other. And so discovery, however, can be driven across communities. And indeed, it was. The FOP mutations were discovered in 2006. That data could inform the DIPG efforts that really began in 2011, essentially. Please email, and I'll try not to talk as long about the merits of it and really just focus on how we can potentially support your own efforts in building a resource. It's incredibly valuable to have domain-specific efforts leverage these types of initiatives. And the NIH, who's sponsoring the Kids First data resources, is highly advocate of exactly that approach.
Great. Hopefully you have time for one last question.
Yep.
And the person who asked that question about connecting responded exactly our thoughts, and we're very excited to collaborate with you. The last question is from Emily Pickle, and it says, since regulatory work takes so much time, has the CBTN considered centralized regulatory, example, central IRB?
Yeah, so we actually have a central IRB of record, and it's absolutely right that it takes a lot of work and effort. The NIH actually now requires a central IRB for NIH-funded initiatives exactly for that reason. So all of the institutions within the CBTN essentially follow the same SOPs, same consent workflows moving forward. There's still existing challenges in the IRB process in general between institutions, because not all IRBs interpret things equivalently. But this is where patients and communities can be transformational. Informed patients can drive the use of their data and specimens in ways that are essentially overriding of any interpretive or bureaucratic process driven by a regulatory environment. And more specifically, what the Office of Civil Rights really made clear over the last couple of years is that patients actually own their clinical data. Patients own their healthcare in ways that we as a community need to do a better job of empowering them to inform how they want that data to be used and or specimens empowered. And really, this is a space where we're spending a tremendous amount of time and effort to further develop these portals and environments such that they not only support a researcher, but ideally, right, the vision ought to be that they fully support a patient's own need to interpret and understand their disease. Now that's a tall order, but it's not impossible. And what we find is that the minute you're diagnosed with a rare disease, many families either become experts or connect to expert families and communities. And so they come in asking very specific questions that we hope portals and environments like this can support them. That's a long way of saying that there's still work to be done on optimizing around the regulatory process. But even the NIH, and it's certainly true in the EU with the implementation of GDPR, have come to recognize that patients ultimately should be informed and empower their data and specimens in ways that are aligned with mission. Namely, every patient that we know wants to accelerate the discovery process associated with their child's disease.
Great. And Emily responded, brilliant solution, agree entirely. Families are powerful and this is solvable. So with that, that's the last question, Adam. I want to thank you again for doing this lecture today. And I want to thank everyone for joining us. And hopefully together we can make an even bigger difference for these families.
I look forward to it. Thank you so much for joining.
Thanks, Adam.